Hello. The video you're about to watch is part of the Age Presents video series. This series is produced by the American Aging Association to provide an introduction to foundational and advanced topics in geroscience research. As you proceed through these videos, we encourage you to think about the following questions. 1. What relationships between aging and disease are described in the video? 2. What anti-aging interventions are proposed? And 3, what model systems were used in the research and why? We would like to thank our sponsors, the University of Washington Healthy Aging and Longevity Research Institute, the Jackson Laboratory Center for Aging Research, and the Nathan Schock Centers of Excellence at both of these institutions. If you are not yet a member, we encourage you to visit our website and become a member of AGE. AGE members receive free access to our Society Journal Geroscience, reduced membership fees for our annual scientific meetings, opportunities to interact with leaders in the field, all of the lecture materials and slides for this lecture series, and much more. In addition, we have a highly active trainee chapter, which provides numerous opportunities for networking and career development. On behalf of AGE, thank you for watching and enjoy the lecture.
Thank you very much. This is my pleasure to provide this virtual lecture series for the American Aging Association on the topic of cellular senescence and aging. My name is Darren Baker. I have a research lab at the Mayo Clinic in Rochester, Minnesota, and I will be talking today about a number of reasons why I think senescence plays a causal role or contributes pretty significantly to various pathologies with age. And thank you for your attention. I always start every presentation with this disclosure statement. A number of things that we've done have generated a lot of interest in the space of developing drugs to potentially go after and eliminate senescent cells, and I'll tell you maybe why that's relevant as we go through. So we have a partnership with Unity Biotechnology that has licensed a lot of our technologies for usage. And so basically I include this just to be safe. Not everything that I do is kosher by the Mayo Clinic conflict of interest policies, but again, I always include this. So I think about age-related diseases maybe a little differently than others, where I think about age-related disease more as a consequence of our success over time. With any organism, especially if we're talking about mammals, there are two parts of our lives that are not negotiable. There's our period where we're born and our period when we die. And those are pretty much, again, things that we can't control. There are various phases of our life. We think about, if I think about human biology and break it into these periods, there is the age between when we're born and 15 years or so of age, when our pre-reproductive period of life, followed by our reproductive active space of our life from 15 till about 50, and then our post-reproductive period of life. Now, this reproductive period can be extended, as we know, but generally this is pretty safe to say that your reproductive activity after 50 is pretty minimal. If we think about history and more in the context of how evolution works, if we think about genes that would be really selected for in the natural selection process, there's really heavy selective pressure early in our life, where if we have a particular genetic mutation that would be really negatively influencing our health, that would be something that theoretically would be selected out of the population and not get propagated onto offspring. So once we start being reproductively active, though, those genes that might have deleterious effects, we would not be selecting out. So those will be maybe propagated within the human genome, and there may be actually the accumulation of genes that may be in fact causing our death during these later phases of our life. I think this age of 50 is very relevant because in the year 1900, that was the expected life expectancy of a human, was 50 years of age. So just 120 years ago. That is prior to a lot of public health improvements that have definitely influenced this time of death, where there's things like clean water, sanitation, other health measures, working conditions, treating communicable diseases that have really influenced this to push us well past this age of 50 to now into our later 70s for both males and females. But what I think is interesting is that if we look at age-related diseases, age-related diseases really start to accelerate after about this 5th decade or so of life. And some examples of these age-related diseases are things like Alzheimer's disease, arthritis, atherosclerosis, et cetera, et cetera, et cetera. And what my lab has become interested in is really understanding if we can influence the rate of aging, can we maybe influence the rate of the development of all of these pathologies at once? So the idea is instead of just going specifically after a causal effect driving Alzheimer's disease, if we influence aging in general, is that sufficient to kind of delay out these various features? Part of this is also a concept of the geroscience hypothesis that has been really championed by Felipe Sierra of the NIA, talking about the idea of these accumulation of age-related diseases and again, influencing the rate of aging to attenuate many of them. So again, the idea would be to probably to work on trying to improve health by slowing aging. I think it's relevant because most of what I work with is animal models, and many of our animal models have been developed with particular mutations where we're really trying to understand what those genes are doing, where we're trying to understand kind of the biology behind those particular genes without many real understanding of how those genes are influencing more in the context of aging biology. So genetic knockouts, for example, those things that maybe are germlines, those are from the beginning period already having this particular gene knocked out. What relative to if we were to look at this in a later age, what might be the influence of these particular genes in late life? There's some evidence that aging, in fact, can be delayed, or at least there's suggestions that evidence that there is an ability to attenuate the rate of aging. A number of people are working on really understanding, taking the human population, really studying these people that are extended longevity individuals that live to excessive long ages, over 100 years of age, which are called centenarians. One example of this is this woman, Jeanne Calment, a French woman that lived to be nearly 122 years of age. Interestingly, if you look at her behaviors, she was doing a number of things that would not be thought of as really promoting a long life, including smoking for over 100 years of her life. But looking in these centenarians, a number of people have found particular SNPs or other genetic alterations that seemingly correlate with this ability to have exceptional longevity within humans. Something that we've done within laboratory models for a long period of time now is caloric restriction, where if we reduce the amount of caloric intake in laboratory mice or many other species, you can extend the lifespan. In mice, you can get about a 20, 30 to 40% increase in lifespan of these animals. And not surprisingly, this increased lifespan also is accompanied by an extension of their health span, meaning the time that they're exhibiting really no pathologies of age or living healthily. There's a large group that's interested in trying to identify compounds or drugs that may be able to alleviate symptoms of aging or features of aging. This is part of a whole cohort of studies that are done in collaborative work between Michigan, Texas, and at Maine at the Jackson Labs, doing these extension studies or looking for if there's an impact with various drug treatments. And 3 examples are shown here that seemingly have some impact on the rate of aging by doing drug interventions. And lastly, one that I'll talk about is an interesting studies that have been done with heterochronic parabiosis, which are studies of taking old individuals and basically stitching them to a young individual such that they have a shared blood supply. When you do this, this is something that you can do in laboratory mice. It's not so easy to do this, to translate this into a human. But what's interesting is that you see that there are factors that are within the young circulation that have beneficial impacts on the elderly pair in this particular heterochronic parabiosis experiment. But this comes at the detriment of the young individual. So there's also factors within the old circulation that are detrimental to a young individual. Many people are working on trying to replace this heterochronic parabiosis now with doing things of doing plasma instead. So that would be something that would be more translatable into the human condition, where you can maybe get young factors or rejuvenating factors that would be coming from a young individual that can be transferred then into an elderly individual. There's a number of aging theories about why, in fact, we do age. Some of these are there's hints that each of these may be having some role in the aging process. One of them is a cross-linking theory, thinking that there's unwanted cross-links between various molecules that are unable to be resolved, that increase over time and cause dysfunction of cells. Another is related to a genome maintenance theory of aging, where DNA damage is accumulating over time from either things within our food or within intracellular processes that cause errors or other issues within DNA, that is unresolvable and you have problems with your genome, and that ultimately leads to age. Similar or kind of related idea is a free radical theory of aging, where there are reactive oxygen species or other byproducts of metabolism that are actively causing damage to other molecules, be it DNA, proteins, or other effects within the cell. But what I'll focus for my talk on is on the cellular senescence theory of aging. The senescence theory of aging was something that is actually, in the grand scheme of things, a relatively young idea. In about 60 years ago, this person, Leonard Hayflick, in working with Paul Moorhead, was working to identify, or really was trying to culture, human fibroblasts, and had found out that the idea was at that time that human fibroblasts were immortal, that they would grow forever in culture. But what Hayflick found is that over time, there is a spot where after about 50 to 60 population doublings, these fibroblasts then entered into a state that they called senescence, which was now an irreversible growth arrest, where they were still living within the culture. As long as you were replenishing it with fresh media, it would stay alive, but there was no further division of those cells. This became known as the Hayflick limit. So this is something that is now well accepted within our field, that normal cells have this capacity for division to a particular point, and then they reach this limit and are no longer able to divide. It didn't take long for this to then be conceptualized to underlie the aging process within individuals, where the thinking is with advancing time, we get to a spot where our cells are no longer able to divide and replenish themselves, and it leads to this idea of increasing organismal age with time. In later studies, it was found that really the important regulator of this senescence effect, at least in vitro in fibroblasts in human cells, was the erosion of telomeres. And when telomeres got critically short, cells were unable to divide any longer. So this is kind of the molecular understanding for why the Hayflick limit is ultimately reached. The idea that senescence maybe played a role in vivo, though, was still very unclear, being as we didn't really have great tools for identifying the presence of senescent cells. Despite her small stature, a giant in the field of cellular senescence research is Judy Campisi, who's based at the Buck Institute in California. Judy has done a variety of studies that have been really instrumental in really trying to understand what senescent cell biology is actually doing, both in vitro and in vivo. But this particular study from now 25 years ago in PNAS from Judy's group talked about the accumulation of a particular phenotype known as senescence-associated beta-galactosidase activity. The idea is that this is an enzymatic activity assay that the lysosomes of senescent cells at a particular pH have a particular activity against this particular substrate. And what you find is that when cells are labeled, meaning that they're going through DNA replication, there's very few cells that are beta-gal positive, SA beta-gal positive. But as you have a reduction in the amount of labeling of nuclei, meaning that there is not cell cycle progression taking place or DNA replication, you see an acceleration or an accumulation of SA beta-gal positive cells, where many cells now are becoming beta-gal positive. You can use this same activity assay in vivo and look for the change of cells to develop this colorimetric assay of becoming blue in tissues when they're becoming senescent. This is from a young individual skin, this is from an old individual skin, and you can see the accumulation of these beta-gal positive cells, both in epidermis and dermis is in this particular study. So we now had a tool where we could at least characterize, do senescent cells accumulate with advancing age? And it was found, in fact, that senescent cells do accumulate with aging and at sites of age-related dysfunction. So the thinking is that when we're young individuals, we may have senescent cells that will show up for a particular purpose, but we think that they have a particular purpose why they are induced in that spot. Maybe it's in response to a particular type of damage. What we think is that as we age or as we develop pathologies, more of these cells will accumulate to a spot where when we are looking in old individuals, we always will see an increased burden of senescent cells relative to a very young individual. This is pretty consistent amongst mammals, pretty consistent amongst different laboratories as well. So this led to the idea that maybe these senescent cells, not only are they there, but they may be causal and implicated in driving pathologies with this increasing age. There's a number of different stressors or factors that can induce the senescence program. This is just a few examples of this. Many of these stress signals are things that would be actively trying to promote transformation of a cell or causing dysfunction within a cell. And the cell will then respond to that by activating pathways to try to ameliorate the damage. Many of these involve cell cycle regulators that are very important tumor suppressors. One of the arms of this pathway that senescence engages is through the p53 pathway. The other is through the p16 pathway. Both of these, if you knock out either p53 or p16 in laboratory mice, those mice are tumor prone. So it underlies the importance of these factors implicit in preventing neoplastic transformation of cells. But the idea is in response to maybe something like excessive DNA damage signaling, if you're irradiating cells, you get activation of these pathways to engage a cell cycle arrest. The thinking is that this cell cycle arrest is first trying to ameliorate the damage, activating different repair pathways that can then attenuate the stress signal that's coming from these cells. But if the damage is too severe, the cell is not, it can go through a spot of going directly into an apoptotic cell death if the damage is too severe. But if the cell has first engaged this transient arrest mechanism, engaging an apoptotic response from that spot is very challenging for the cell. So instead, what the cell has done is it now goes into this state known as senescence, where we think it's a long-term stable growth arrested state of these cells. They develop a variety of phenotypes. One of them is the acquisition of this SA beta-gal positivity. However, not all senescent cells are beta-gal positive, nor are all SA beta-gal positive cells senescent. So there's a little bit of problem, but generally, many times we see acquisition of a beta-gal positivity in a senescent state. Importantly, senescent cells are also thought to then influence other cells in the environment through factors that they're secreting. This is collectively referred to as the senescence-associated secretory phenotype that consists of a variety of cytokines, chemokines, matrix metalloproteinases, other growth factors, things that are influencing other cells in the environment. So although we think the burden of senescent cells may be quite low within tissues, the thinking is that those limited number of cells that are becoming senescent really can influence a lot of cells in their environment. From a cell intrinsic mechanism, the establishment of senescence is a very potent anti-cancer mechanism. So the idea is if you have a particular type of oncogenic activation or one of these other cell stress signals, if you now promote that cell to go into a spot of becoming irreversibly growth arrested, it will not become neoplastic. So in a way, that's a very potent tumor suppressive function. But what we think is that this comes then at the consequence of driving aging and other age-related diseases. The idea about the senescence-associated secretory phenotype is also not that old. So this is some work from about 12 years ago, again from Dr. Campisi's lab, talking about looking at what were the secreted factors coming from different types of senescent cells induced to become senescent from different types of stress signals. And largely what they found was that there was a cluster of genes that were upregulated and expressed and secreted within senescent cells that we then started to think about these cells that are now becoming senescent are now actively communicating with other cells in the environment. And some of these pro-inflammatory signals are things that we see increasing with natural age that may be then underlying some disease-related pathology. Where it becomes complicated is that some of these factors, things like IL-6 expression, for example, We've seen that there are beneficial effects that they can recruit different types of immune cells to the environment to potentially eliminate senescent cells. But we also know that there's this two-sidedness of IL-6 where it can be also deleterious and ultimately promote tumorigenesis as well. So the SASP is complicated. It's a lot of work that we're still trying to really understand what the senescent secretory phenotype looks like in vivo, because a lot of our studies have really relied on the use of tissue culture systems for really identifying and understanding what this phenotype looks like. Early, the idea about senescence and how it may play a role in age-related disease or dysfunction with age relied primarily on the cell intrinsic mechanism of the thinking that if you're driving a senescent state in something like the stem cell, that would cause a phenotype reminiscent of a stem cell exhaustion phenotype, where there would not be cells to replenish tissue upon damage or with age. But what we subsequently have learned is that the acquisition of senescent cells in the environment through the nature of the senescent secretory phenotype can lead to dysfunction of stem cells. So in a way you have a similar appearance, almost like a stem cell exhaustion effect. We know senescent cells can actively drive destruction of the extracellular matrix and promote degeneration, as well as tissue fibrosis, as well as driving things like a chronic inflammatory state within the tissue. Through the secretion of these inflammatory cytokines, immune cells will be recruited, and we think that this is underlying many aging and disease pathologies. Now, how did I become interested in senescence and aging? Ultimately, the answer is it was through a failed experiment, if you want to look at it that way, where early in my career, I was working to try to understand cancer and really working to understand this particular phenotype that cancers develop. This is a mouse tumor, where if you look at these tumors, they have an abnormal number of whole chromosomes, a condition known as aneuploidy. And what we were doing was we were trying to develop and design mouse models to get this aneuploid state to ask, is aneuploidy sufficient to drive tumorigenesis? Pretty simple, straightforward question. Again, it was kind of an old question in biology. Now it's about 140 years old question within biology about this idea of aneuploidy driving tumorigenesis. But the thinking was, can we develop mouse models to get this chromosomal instability phenotype to maybe get aneuploidy alone, the accumulation maybe of 2 extra chromosomes or maybe even 1 extra chromosome. Maybe that chromosome has a lot of oncogenic activity, or maybe even just protein imbalances that are taking place as a result of aneuploidization is sufficient to promote tumorigenesis. Or you can think of similarly if you had an aneuploidization where you lost 1 entire chromosome that had a lot of tumor suppressive effects, that you would also see a tumorigenesis phenotype. So we developed, and the molecular model that we used was making mice that expressed low amounts of a mitotic checkpoint protein called BUBR1. I'm not going to go into detail about what BUBR1 does, but it essentially makes sure that during the process of cell division in mitosis, that metaphase is delayed until all chromosomes are properly aligned in the metaphase plate, so that you get equal distribution of chromosomes into 2 daughter cells. If you attenuate or mess up that process of that surveillance mechanism of the mitotic checkpoint, you drive this aneuploid phenotype. And what we found is when I first started in the lab, I got handed a box of mice of these BUBR1 hypomorphs and was told to kind of figure out what it was that was actually happening, because all that we knew was that these mice were dying very early in their life. In the early 2000s, there were a number of different animal models that were being published that were part of DNA damage models or mitochondrial mutator models that talked about progeroid or segmental premature aging phenotypes that exhibited phenotypes within laboratory mice. And so we started looking at our animals, and we found that there were a number of features of premature aging that were accumulating within this BUBR1 mutant mouse. One of them was the accumulation of bilateral cataracts. All of our mice, 100% of them, have bilateral penetrance of cataracts within about 8 weeks of age, a very striking phenotype. We also saw that they developed this kind of abnormal rearward curvature of their spine, which we call lordokyphosis. that we looked for whether this was due to an osteoporosis effect or a skeletal muscle atrophy effect, and we largely found that it was underlied by a skeletal muscle atrophy that was accumulating within these BUBR1 hypomorphic mice, driving this dysfunction. We also found that these animals had very low amounts of adipose tissue, and in early experiments, when we took the adipose tissue from these BUBR1 hypomorphic mice, we saw that they had very high levels of senescence-associated beta-galactosidase activity. We also looked for markers or basic evidence that this pathway of senescence was engaged in these BUBR1 hypomorphic mice, and we found in these three tissues that there was evidence that the P16 pathway was elevated, suggesting that there may have been an accumulation of senescent cells within this particular model. In an experiment, but ultimately what we were trying to do with these BUBR1 hypomorphic animals was to get them to get tumors. That was the design of the experiment, was to get aneuploidy and drive tumorigenesis. These mice did develop aneuploidy, so we were doing what we wanted to do for the experiment, but unfortunately these mice were dying from their premature aging phenotype, too early developed phenotypes of tumorigenesis. So what we thought was, what happens if we now develop this aneuploid state and we couple it with some tumor suppressor knockout animals? The idea being that those tumor suppressor knockout mice will get cancer. Does aneuploidization now synergize to develop aggressive tumors in that particular model? One of the mice that we made was a mouse that had a knockout of P16. So as I mentioned, P16 knockout animals develop this cancer phenotype. But what we found is when we had now made mice that expressed low amounts of BUBR1 and no P16 expression, we saw an attenuation of these age-related pathologies in the eye, skeletal muscle, and adipose tissue. So we were able to really delay these aging phenotypes by not allowing the animals to express P16, but that came at a consequence to these mice where they were now more tumor prone. So in a way, the experiment was successful to demonstrate that aneuploidization and the loss of this tumor suppressor gene synergized to develop tumorigenesis, but the aging phenotype became extremely interesting for us to really try to explore. I talk about the idea about cell intrinsic and cell extrinsic effects of senescence and why we've gone into different phases about senescence and the biology behind them. I use this figure of Ned Flanders, who's the neighbor of the Simpsons, to talk about the idea about how senescent cells can influence other cells in their environment. And there are some good aspects about the accumulation of senescent cells that have been talked about in recent years, demonstrating that senescent cells are important for at different stages of repair processes or in the process of development, where the acute acquisition of a senescent cell is actively driving some type of repair process, something that is more in a state of trying to have benefit to the organism. And I think about senescence in that way more in the acute state, where it's induced at a particular point in time for a particular purpose. What we've started to really understand is the idea about how senescent cells can also be detrimental, or the bad side of senescence, where the accumulation or the long-term stabilization of senescent cells in tissues has been shown to drive different phenotypes associated with aging, whether it's through tissue fibrosis, promoting tumorigenesis, this chronic inflammatory state, other types of dysfunctional tissues, where the thinking is that the accumulation is more in the idea of a chronic long-term senescent signature negatively impacting cells in the environment. So there's this kind of two-sidedness, similar to the idea of senescence arrest in the first place, where it's good for preventing tumorigenesis, but it may be promoting the process of aging. So the thinking was, how can we keep this good side of senescence intact, but then subsequently take away the bad side of senescence? And that led to a model that we developed now almost 10 years ago, more than 10 years ago when we were developing it, called the INCATAC model system. There's other models similar to this, where the idea is that you are using different drugs in a genetic context of an engineered mouse model to eliminate senescent cells. In this case, we used a small portion of the P16 promoter to drive the expression of a drug-inducible caspase-8 molecule. As cells were entering into senescence, they would start to be expressing P16. They would also theoretically start to express this transgene, but nothing would happen to those cells because the caspase-8 is in an inactive state. The addition of an artificial drug known as AP20187 causes the dimerization of these caspase-8 molecules in a senescent cell. The dimerization of caspase-8 subsequently leads to its activation. The activation of caspase-8 is thought to then contribute to apoptosis, and then the thinking would be is that we would then selectively eliminate senescent cells with the addition of this artificial drug when a cell has now entered into a senescent state. So you keep the arrest mechanism intact, all of the stress signals or whatever may be underlying why a cell is becoming senescent would be intact, and now we've subsequently made that cell amenable for elimination through the administration of this drug. That was essentially the concept that we developed and designed and tested. And the first model that we tested this in was in the BUBR1 hypomorphic mouse. We're still working to try to understand molecularly why BUBR1 hypomorphic mice develop senescent cells in the first place. But in this case, what we've seen is that we have animals where we treated them from weaning age on, starting after about 3 to 4 weeks of age in a BUBR1 hypomorphic mouse. This mouse has been receiving vehicle until 10 months of age. It develops those pathologies that I talked about earlier of the kyphosis, the low amounts of adipose tissue, the skeletal muscular atrophy, the cataract phenotype. And what we found is that when we treated these mice with this AP drug consistently, so we treated them throughout their life, beginning at weaning age, we're able to attenuate these aging phenotypes in this BUBR1 hypomorphic mouse. So the idea of skeletal muscle atrophy, these other effects were then attenuated by removing their senescent cells with this drug treatment. We subsequently have done a study now, I guess more than 5 years ago, where we treated normal mice. So unlike the BUBR1 hypomorphic mice, it's quite easy to make a laboratory mouse sick. You can mutate hundreds of genes and make a laboratory mouse sick, but it's much more challenging to make a laboratory mouse healthier. In this case, we used the same Incotact construct. We put it just into a wild type mouse, and we started treating mice really prior to a large accumulation of senescent cells at 12 months of age in these mice. And we treated them again constitutively from 12 months of age until a late life to see what the impacts of senescent cell removal may be with these mice. If the video plays, there's an animal in the foreground here that is AP treated again, starting at 12 months of age. These are 28-month-old mice. And this is its vehicle-treated partner. That's actually a litter mate control. And what we see is that there's obvious improvements in terms of the overall appearance of these animals are improved. These mice are able to exercise on a treadmill much better. You can also see that there are some improvements in coat color. And we published this story where we saw that there was about a 25% or so extension of lifespan in these mice. Primarily, we think that the extension of lifespan in this model was because of an attenuation of the tumor phenotype. These same mice that were treated got tumors at the same sites as the mice that were vehicle treated. It's just that they got them later. So we think that may be underlying why they have this attenuation of the lifespan in the model that is vehicle treated, is that it's developing tumors a little bit faster and that the senescent cells are promoting tumorigenesis in these mice. But importantly, we saw a number of aging phenotypes that were attenuated with senescent cell removal. We saw no detrimental impact in the context of laboratory mice in a controlled environment for senescent cell removal, where many of these aging phenotypes are attenuated now with senescent cell removal. We're trying to do a little bit more further study in this space. When we published this story, it kind of led to the idea that maybe, again, senescent cells and age-related diseases may be related. And there's a number of things that we think about universal aging traits that pretty much are not negotiable. As you age, a variety of these factors or these traits will start to accumulate within populations. We right now don't really have a good understanding of how much senescent cells play a role in these processes. We're still working on trying to understand these kind of universal aging traits. But about 10 years or so ago, we started to think about where have senescent cells been shown to accumulate with age-related diseases? And there's a variety of different tissues that have demonstrated some hint that senescence might be playing, or at least contributing to the pathology, or it's accumulating with pathology in a variety of human conditions. Since the development of these mouse models that allow for the elimination of senescent cells, we subsequently have been able to find many of these diseases where we've been able to demonstrate a clear causal link between senescence accumulation and pathology, where when we treat these animals to remove their senescent cells, we have an attenuation of many of these features of aging. If they're even, maybe it's not even necessarily a disease, but cataracts, for instance, is something that we saw an attenuation of the cataract phenotype by removing senescent cells as well. This is just a snippet of what's been going on. But if you look at the time when many of these are coming out, this is within the last 3 to 4 to 5 years is when many of these studies have been being done. Many of these utilized the approach of the genetic models that we had talked about related to senescent cell elimination through a genetic means. But clearly, that's not something that can be really at all translated into a person. This has led to the idea that is there something from a pharmaceutical approach that we can also use to go after selectively senescent cells for elimination in these age-related pathologies within people. A lot of this work has really been spearheaded from a colleague of mine here at Mayo Clinic, Jim Kirkland, that about 5 years ago, they published a paper talking about where senescent cells may be sensitive for elimination, looking at what pathways are really active to try to keep a senescent cell alive. And they referred to these as SCAPs, or the senescent cell anti-apoptotic pathways, where the expression of these pathways is thought to try to basically keep a senescent cell around. And the thinking is that if you're attenuating these anti-apoptotic pathways, the cell is so damaged that it may be sufficient to promote that cell to drive it into a spot of dying through a pharmaceutical intervention. So this would be something that can be done in the clinic today. And in fact, there are many clinical trials that are being done with these senolytic drugs going after trying to selectively eliminate senescent cells. There's also a different category of cells or of compounds known as xenomorphics, which xenomorphics are more designed to attenuate the effects of senescent cells. If we're to believe that the senescent cell phenotype of being really pro-inflammatory or the secretory phenotype is underlying the causation of disease, xenomorphics are trying to attenuate the secretory profile largely of a senescent cell, such that you still would have a senescent cell around, but it just would not be excreting some of these negative compounds that we think are really influencing negatively other cells in the environment. Interestingly, Dr. Kirkland's group has developed a different variety, and many others, in fact, have developed different ways of assaying for senescent cell sensitivity to various senolytic agents. Interestingly, there seems to be some selectivity where not all senescent cells or different ways of entering into senescence make cells sensitive for different senolytic agents, as well as the thinking that these drugs or these treatments can be more given intermittently and not necessarily need to be constitutively present within the system to improve function, both in progeroid and geriatric mice. A number of the pathways of senolytic molecules are just illustrated here, and it's almost hard to stay up to date because there's so much active work being done in this space for the development of new molecules or new ways of intervening with senescent cell biology. This is looking specifically at the senolytic molecule pathway. There's a couple here, dasatinib and quercetin are the ones that have really been piloted from Dr. Kirkland's group. Those are really heavily being, at least there's a number of different studies in the clinic where dasatinib and quercetin, either alone or in combination, are being utilized to see if there's any attenuation of pathology within specific age-related diseases where there really is no treatment available for those particular maladies. One of the drugs that we use in my laboratory frequently is ABT-263 or navitoclax. The thinking here is that this is a drug that really counteracts the pro-apoptotic, or there's the pro-apoptotic machinery of BCL2, but with the idea of navitoclax, you then suppress this anti-apoptotic faction to drive to the senescent cell to become now apoptotic through intrinsic means that are being elevated. And again, many of these others have been talked about. Something that is some interesting technique and technology relates to the acquisition of galactosidase prodrugs, where the idea is that you would utilize or do a linkage between something like a nanoparticle that would be galactosidase containing, that in a cell that would become senescence, or that in a cell it would be senescent, the lysosomal activity would then subsequently lead to the activity of that particular senolytic agent to then get delivered to the right cell to kill that particular senescent cell. Because as we know for navitoclax works through this mechanism of killing cells, but that works both in the context of senescent cells and in non-senescent cells as well. So there will be cells like platelets that will be sensitive to elimination through navitoclax exposure, that through the administration of these navitoclax galactosidase prodrugs, the thinking is that now you get delivery to the site where it needs to be active, into the senescent cell, where it will then become active and subsequently trigger senescence within that, or trigger apoptosis within that senescent cell specifically. The last topic that I'll talk about is xenomorphic molecules. Again, this is just a little bit of a list of some of the various compounds that have been talked about to influence this senescent cell secretory phenotype of senescent cells to really modulate the pro-inflammatory state largely of senescent cells to attenuate this rate of kind of chronic inflammation that's being driven within tissues with advancing age. Again, lots of work is being done in this space for the development of senolytics and xenomorphic drugs. My lab is very focused on basic biology of senescent cells in terms of where they're accumulating, why they're accumulating, what they might detrimentally be doing. And it's really nicely then dovetails with the development of some of these drugs where we can do things of our genetic means of elimination and then look for validation with different senolytic or xenomorphic drugs to really understand, is it the senescent cell themselves, that it's this irreversible growth arrest that might be contributing to the pathology, or is it really the pro-inflammatory signaling that's driving this pathology? With that, I'll finish with some acknowledgements. I apologize for any person that I was unable to include for the presence of time on talking about their work. There's been, again, a lot of development in this space over the last even 5 years or so. My group, as well as others at the Mayo Clinic that we work in close collaboration with, as well as external collaborators, including specifically Jan van Dersen, whose lab I was in when we were really working on generating this INCATAC model. And it's a great group that we've been working with, including people like Judy Campisi, because without people like Judy or others, you really don't feel like you really even know what you're talking about senescent cell biology unless you really have her on board. With that, I will conclude. Thank you for your time and your attention for this. If anyone wants to reach me for questions, I am not terribly difficult to find. And with that, thank you very much.